Hey all, so in this video I'm going to discuss how you need to prepare for this agriculture biotechnology syllabus. Okay, so many of you, I think you had applied for this ICAR NET, which is also called as ARSB NET examination. So in this video I'm going to discuss you the syllabus for code number 1 agriculture biotechnology. So those children who selected this particular code, I think this video will be really helpful. In the video I'm going to tell you which topic is important, where you need to prepare and how you need to prepare. So first is unit number 1, which is cell structure and function, which is just equivalent to, which is just similar to unit number 2 in case of your CSAR syllabus. Okay. So first for this prokaryote and eukaryote, you need to refer Prescott book. Next for the rest of things like plasma membrane, cell wall, structure of various organelle, cell division and cell cycle and protein secretion and targeting, and how a cell has been growing and differentiating. For all these topics, you can refer Molecular Biology of Cell by Albert. Okay, I'll be giving the link of all those books in the comment section or in the description of this particular video. Okay, so for this particular unit number 1, you can prepare from this particular Prescott and Albert Cell Biology book. Next is unit number 2 and unit number 3, sorry, which is similar to your unit number 1 in CSCR syllabus. So with regarding to unit number 1, if you're already having a biochemistry notes which you have taken for your BSc, MSc or even for CSCR examination or for some other examination, just utilize that particular note alone. No need to prepare any special biochemistry notes with regarding to this particular ARSP NET examination. At the same time, you need to give a special focus for plant biochemistry. Okay, so instead of studying a general biochemistry, just go for referring few plant biochemistry notes. Okay, it will be really beneficial for you. But nearly 90% of questions will be coming from general biochemistry only. Okay, so give a special focus for plant biochemistry. Next is enzymology. So with this regarding to this particular unit, you need to have a brief understanding. You need to know about the structural conformation of various enzymes, how enzymes are being classified. Okay, and also you need to know about how an enzyme will be isolated, purified and characterized. You need to know the entire step, and you also need to know about enzymatic assays that are used in the field of biochemistry. Okay, next you need to know about the mechanism of action of various commonly employed enzyme. Okay, so with regarding to this particular mechanism of action, you can refer Pathfinder book. Okay, and if you ask me whether Pathfinder book alone is sufficient for clearing this ICAR NET exam, means no. In Pathfinder they had given only basic understanding. After reading the Pathfinder, you need to go for referring few textbook. Okay, because with the Pathfinder notes alone you can't able to clear this ARSP NET examination. Okay, next I had given about immobilized enzyme. So steps and what are the different methods available for immobilized enzyme, and also you need to know about the applications of immobilized enzyme, and you need to know about this protein engineering. Okay, which is very important topic. Next is molecular genetics, which is similar to unit number 4. Regarding the unit number 4, starting from concepts of gene and prokaryotic and eukaryotic chromosome and how a prokaryotic gene is organized and eukaryotic gene is organized. So just give a general understanding. So for which you can go for James T. Watson book. That is sufficient. Additionally, even though you didn't prepare for these topics which I mentioned over here, just prepare notes for mutation topic, recombination, site directed mutagenesis, transformation, transduction, conjugation, transposable element and transposition, from which you can able to expect at least 5 to 6 questions coming from mutation, transposon, site directed mutagenesis and this particular gene transformations techniques. Okay. Next is unit number 5, which is again a gene expression in which they have given notes about transcription, for which you can refer James T. Watson book alone. Okay, so you need to know about operon concept, like you need to know about lac operon, trip and ARC operon. So that particular thing is very important. At the same time, you need to know about enhancer, where it is located and about the mechanism of enhancer, activator, repressor, co-activator and about co-repressor in prokaryotic system. At the same time, you need to study about eukaryotic system, and you need to know about the promoter elements like Shine Dalborough sequence. What are the sequences that are present in promoter, both in prokaryotes and eukaryotes. Next, you need to read about this post-transcriptional modification, how protein has been transported to various locations location, like that you need to know. And also you need to study about the genetic code. Okay, so from genetic code, at least 2 questions will be coming, starting from the person who determined the genetic code till you need to know about which particular amino acid have many code, like that you need to know about stop code, a start code, and some other unusual codes. Okay, so for which you can refer any molecular biology book, but my personal choice is regarding to this James T. Watson. Instead of going for referring genetics book, refer molecular biology book more with regarding to ARSB NET examination, because in ARSB NET examination, that is specifically for Agri Biotech, from genetics, that is I think in syllabus they didn't mention anything regarding to genetics with regarding to your pedigree, like that. Only we got a thing like transformation, transduction, conjugation, like that only we got. Okay, so just refer molecular biology. From molecular biology, you can expect a huge array of questions. Next, you need to know about the translation and about the control of translation and post-translation modifications. Again, very important thing. You need to know about the post-translation mechanism, how it been undergoing, and about the location of those post-translation modification. Next important topic is molecular biology techniques. Okay, so you need to know about all the techniques. Don't go into detail, just you need to know about the technique, principle, and application. That alone you need to study. Give more focus on chromatography, electrophoresis techniques, and immunology related techniques. Okay, next is with regarding to gene cloning. Okay, so this is also again an important thing regarding to gene cloning. First, study a generalized concept of gene cloning and go for gene cloning in plants. Okay, so how gene cloning is being done for plants. Okay, next is regard you need to know about restriction enzyme types, for which you can refer analysis of genes and genomes book. Okay, I'll be giving the link of that particular book. It is Analysis of Genes and Genomes by Richard Rees. Okay, and you need to know about, or else you can download these notes. Okay, so for example, these are e-notes that are available in online. These e-notes are being published by either by agriculture universities or those institutes that are coming under ICAR or some foreign universities. So each university they have a course titled Principles of Plant Biotechnology. So for which they will be preparing a e-notes from a standard textbook. So in this particular, see, this is a e-notes from Dale University. If you want, I will be giving the link of this particular notes. In that you can able to cover up nearly 75% of your Agri Biotech syllabus. You can able to find the tissue culture technique, somatic hybridization. Here you can able to find the restriction enzymes, vector for gene transfer, and also PCR. Everything you can able to find. Okay, so you can refer for these kind of e-notes that are available either in State Agriculture University website or Centralized Agriculture University's website, or those institutes that are coming under ICAR. If I could able to collect the e-notes for all those things, definitely I'll be sharing you friends. Okay, so you need to know about the types of vector, the vector size, binary vector, TI plasmid, size of each vectors, expression vectors, like that you need to know about all those things. And also you need to know about this chromosome walking and basics of animal cloning and the risk assessment and IPR. Okay. Next is regarding to unit number 8, which is molecular biology. So it is similar to the unit number 6 I had mentioned over there. So in that already we will be studying translation that unit. So additionally you need to know about the structure of ribosome and function of ribosome. And also you need to know about mRNA processing. And additionally you need to know about this ribosome technology, gene fusion, gene pyrimidine, biofortification, and bioprospecting. So with regarding to bioprospecting, biofortification, gene pyrimidine, gene fusion and all, instead of referring few standard textbook, what you should do means, if you type simply, for example, say if you are going to study molecular marker, means you can able to find few research article or review article, or sometimes you can able to find few notes available in PDF format that was published by scientists or those professors who are working in Indian Council of Agriculture Research. They'll be preparing a notes and they'll be putting the notes in the website for the benefit of the student. So you can refer those notes as I mentioned you earlier, or else you can go for any review article. Instead of going for standard textbook, you can go for recent review article which will be giving you entire information. So most commonly questions will also be taken either from a review article, at the same time questions will also be coming from standard textbook. Okay. Next is unit number 9, 10, 11, 12 are highly important. You can able to expect a huge array of questions asking from 4 units. These 4 units, so don't try to skip any topic. First is regarding to photoregulation, phytochrome, mechanism of nitrogen fixation, the biology of stress, transduction, and types of plant hormone. Okay, at the same time, you need to know about this particular cytoplasmic main sterility and what about the nuclear gene that are involved in starch and storage. Okay, so for which you can refer Tay's book, Plant Physiology by Tay's book. In that book, you can able to find this is a standard textbook. Even for CSIR exam also, from textbook only many questions will be asked. Next is for tissue culture. So for tissue culture and all, you can go for referring plant biotechnology book. So here I had given you can able to find a large amount of textbook available for plant biotechnology. So if you search Agri biotech textbook mean you can able to find. But if you put plant biotech books means you can able to find. So I will be suggesting you three textbook, so two textbook that is very simple English only they will be giving and it will be very much interesting also. First is Plant Biotechnology by Slater and next is Introduction to Plant Biotechnology by H.S. Chawla. These two books will be highly beneficial for you. Don't go for any foreign author books. Okay, so only for this unit number 9 you are referring textbook. Apart from that, for this particular unit number 10 and all, you can refer Chawla book. At the same time, you can also refer Slater. It is very much helpful for you. Okay, this tissue culture is very simple unit, or else what you can do means you can refer this particular PDF. Here you can able to find the notes for all kind of cultures like protoplast culture, endosperm culture, embryo culture, anther culture. Okay, so you can refer whatever the note they were very. I have shared you a large choice for you. You can select. Okay, so in this unit you need to study about the different type of cultural techniques. You need to know about application of tissue culture in crop improvement, mutagenesis in case of plant improvement, and cryopreservation technique, and what are the different plant tissue culture repository that are available in India. Okay, and next you need to know about the secondary metabolite protection. Okay, and also applications of those particular things, for which you can refer either the textbook or else you can refer review article regarding to secondary metabolite. Next is plant genetic engineering. So for plant genetic engineering and all, definitely you need to refer a standard textbook, or else if you type plant genetic engineering notes PDF means you can able to find few universities like Annamalai University, Satyabama University. They will be publishing a notes for you. Okay, so if you simply type the heading means you can able to find theory material. These theory material are being prepared by those professors who are working in that particular university. So instead of searching here and there, you can go for referring those particular thing, and this is a very important thing. You need to know about different type of method available for gene transfer method and how a transformers are being analyzed, different kind of molecular analysis, and what are the potential application for plant genetic engineering for crop improvement. And you need to know about abiotic stress resistance, herbicide resistance, and how the protein quality has been improved. Okay, and what about shelf life. Okay, flavor saver tomato, what are genetically modified crops. Okay, so those things are highly important. At the same time, you need to know about biosafety norms and field trial and how transgenics are being released into environment. Okay, and also in recent for the past 5 years, how many transgenic crops or how many genetically engineering crops have been approved for cultivation, like that you need to know about all those things. Recently genetically engineering mustard has been given approval, and currently in India genetically engineering mustard has been cultivated. So you need to know about which particular gene has been inserted and which particular institute is responsible for producing those particular field trial. Okay, the last unit is molecular marker, for which you can refer articles or else few other notes. Those notes are being prepared by ICAR professor who are working in ICAR or few other universities. Okay, because in textbook of course you can able to find, but these molecular markers are very important topic, so you need to know a huge amount those particular molecular markers. Okay, for this particular molecular marker you can refer few article, review article, or else the PDF which I am showing you, that is the lecture note that are available in online platform. Here you can able to find molecular marker definition, brief description about different type of DNA based marker, or several many review articles is also been available in internet. So here you can able to find this review article has been published on the year 2021. Here you can able to find the different type of concepts and methodology of DNA market system. Okay, so these are the important things that you can able to find while preparing with regarding to this agriculture biotechnology. So the next video I'll be definitely sharing you important topics with regarding to agriculture biotechnology. Okay, so thank you friends, thanks for watching this video.